Welcome back. We'll continue our discussion of the chemistry of life, Unit 1. So today we're going to be talking about carbohydrates. So again, a nice shout out to Khan Academy. They have some great AP level articles that you should check out. Some of them go into a little bit beyond the AP level. So make sure you don't have to write everything down, but they're a great introduction. I would encourage you to read it. Everything you read is going to be useful for your understanding what's going on in our course. So when we talk about carbohydrates, this isn't greatly emphasized on the AP exam. However, you do need to know some important terms, just like always. So you need to know the terms monosaccharide, disaccharide, polysaccharide, what those mean with respect to the greater picture of our monomer and polymers in our biomacromolecules. So let's give you an overview. So what are we talking about? Our important biological macromolecules. And we do have a bias towards humans. So we talk more about what are the macromolecules in humans as opposed to plants. Though with carbohydrates, they're actually more, they have a larger role in plants than humans, as we'll see. Okay, so my goal is to keep this video nice and short, tell you what you need to know, not give you too much extra information. So let's go through what are our biological macromolecules we're talking about. Well, these are what are called polymers, many of them. We're going to see one important omission, made up of monomers. And that just means that the macromolecules in our bodies, monomers. Did I spell that right? Let's see. I think so. Yeah, apparently. Why does it look weird? Oh well. Let's just start again. Hello, students. Welcome to our next discussion in the biology of life. So today we're going to be talking about carbohydrates. Carbohydrates are part of our greater discussion of what are our biological macromolecules. We've already talked about lipids and proteins. And what's interesting about a lot of these is a lot of these are actually polymers. So when we say polymer, what we mean is we have a large macromolecule that's important for life that is just a repeating pattern. So DNA, as you know, has this nice repeating pattern that if you denatured it and spread it out, it would just be the same backbone over and over and over again, and then one of 4 base pairs. So it can be represented pretty easily. In a similar way, so that's nucleic acids. Let's write this down. Nucleic acids. And that's both DNA and RNA, are nucleic acids. And we call their monomers nucleotides. And if you're taking notes, I encourage you just write down all of these, just like I did for the top one. So for every polymer, you need to know for the AP test the connected monomer. So that's nucleic acids, such as DNA and RNA. If you're not going to remember that, make sure you write down DNA or RNA. Now, we also talked about proteins. So you should know what are those repeating monomers called that make proteins. So make sure you fill that out in your notes. Quick reminder, it's amino acids. And then also today we're going to talk about carbohydrates. their monomers are called saccharides. And in humans, they're made out of glucose. But in general, you should know both the word glucose, but also the general term, which is saccharides. So just like nucleotides could refer to be something making DNA or RNA, amino acids could be making any proteins in any living thing. Glucose is just a specific one that humans use, but in general, we want to use the word saccharide, which is the Greek word for sweet or sugar. So glucose or sugars. So your body uses the glucose and sugars that you consume to make carbohydrates. Now, other living things such as plants will use other monomers, but they're all called saccharides. So yeah, again, the general term that you should probably write in your notes is saccharides. So all carbohydrates are made out of saccharides, just like all proteins are made out of amino acids, and all nucleic acids are made out of nucleotides. Okay, I think that's all we need to say about polymer and monomers for this class. So let's go ahead and look at the specifics with carbs. Again, nice shout out to Khan Academy. They have a very good article on this, so I would encourage you to go read the whole thing. Their AP Bio page is very helpful. They give you lots of useful information. Not everything's necessarily going to be on the test, but everything is useful to helping you understand. Okay, so monosaccharide is when you only have one. So this is not a long monomer. You just have one sugar floating around. And the most common sugars that we see in living organisms are glucose, and it actually comes in two forms. Well, two forms we see in water. Alpha glucose, which is the more common form in humans, and beta glucose, which is very important for plants forming, which is used in plants to form cellulose, which we'll see. And also there's other forms like ribose and fructose. Fructose you might have heard of from high fructose corn syrup. So that's another one your body can use to break down to get energy. In terms of what your body uses structurally, humans use alpha glucose. Plants actually can use both. If we get two of them together, then it's a slightly more stable bond. And anytime that these form together, they form what's called a glycosidic bond, which I just checked, and I guess you don't need to know that for the AP test. You just need to know this is a type of covalent bond. All of these different polymers are bonded together by different covalent bonds, and based off of the chemistry of it, it's going to be useful for different chemical structures. For example, when you make a very long chain of these, it can be used to make amylose, amylopectin in humans, or it can be used to make cellulose, which is made out of glucose monomers. But again, this is the type of glucose that's actually not so useful for humans. So humans, when we eat cellulose or have cellulose in our food, it just is used as fiber, which is good nutrition, which is good for your intestines because it helps clean them out, but you don't actually get nutritional value out of it. So this is cellulose. Just a lot of these glucose monomers all bonded together with these covalent bonds. And again, this is made out of glucose monomers in the beta form, not useful, not something that the human body can actually digest. As opposed to that, glucose in the alpha form is used in humans in both branched, so see how these make branches, and unbranched forms called amylose and amylopectin. And that's all you need to know. They have different functions. The branches affects how tightly it'll be stacked together and how it's used in your body. But glucose is not just used in its monomer form. In fact, most of the time your body needs to store it by making these long branched or unbranched chains. And we call these, in humans, we call these starches. Okay, that's it. That's what you need to know about carbohydrates. So by now, you should know what a saccharide is. You should know what a carbohydrate is. You should be able to explain what saccharides are most useful for humans and what saccharides are used in plants to make cellulose. And that's pretty much it. So hopefully that gives you a better understanding. Lastly, before we go, I don't know if you could tell from the picture, but carbohydrates only take 3 different elements. Go ahead and make sure, and maybe you can put in the chat or email me, what are the 3 elements that we saw in the carbohydrates? You should add that to your notes. Okay, that's it. Have a good day. Much love. Okay. Now, lastly, we have to talk a lot about nucleic acid because if you noticed in the unit guides, this is emphasized. So the fact that they're doing a whole topic dedicated to nucleic acids, where they only have this one little bullet point on carbohydrates and they don't go into detail, means that nucleic acids are going to be heavily tested. So in fact, notice that unlike carbohydrates, where they weren't really emphasizing the difference between glucose and fructose or anything, they said we need to know the differences between DNA and RNA. And it looks like we do need to know, a lot of the details of the chemistry. So we're going to spend more time on that. Okay, so let's see, what is the essential knowledge? They're similar and different. Okay. Yeah, so we need to emphasize what the difference, which I'll do. And this is the important part. Okay, so uracil thymine, double-stranded, single-stranded. And anti-parallel. That's kind of a lot. Okay, we're gonna have to rely on Khan Academy again to show some nice pictures, because if I'm drawing all of this, no, maybe I'll draw it. I can draw it quickly. Let's try drawing it, and if it doesn't work, it doesn't work. Okay, let's do another recording. Here we go. Let's talk about nucleic acids. No, actually, that might be a really good cartoon or something. Let me check the desktop real quick. If I have any good cartoons, that'd be a fun way to start it. That is a funny one. Maybe we'll start with that one. Anything else? Not easily accessible. Okay, we'll start with this one. Okay, welcome to our last episode, just learning session about the biology of life. Now we get to talk about the most important topic, according to at least College Board, nucleic acids. I would agree it's very important. Fortunately, many of you already know a lot about DNA, RNA, and how it all works. Now, to start, I want to show you a cool cartoon from last year from our biology class. Check out this cartoon. So here we see DNA institution breaking apart our DNA so that these enzymes can actually break apart DNA and do DNA synthesis. So pretty funny. And this one is, of course, crying because he's being torn apart, but that one DNA will now be used to make 2 more DNA. It's for the greater good, Jerry. What a funny cartoon. Okay, hopefully you enjoyed that. So what are these DNA that are constantly being reproduced and used in your body? And is that the only nucleic acid? Of course, as many of you know, when we talk about nucleic acids, there's actually 2. There's DNA and RNA. We're going to talk about them right now. So this is our last lesson on our last big macromolecule, nucleic acids. Now, there's actually other types of nucleic acids, chemically speaking, but biologically speaking, there's just 2 that we focus on, and that is our nucleic acids, DNA and RNA. Let's make the title a nice dark blue. DNA and RNA biologies, genetic information. And just these 2 types of molecules are used by all living things, whether we're going from talking about the tiniest microorganism or whether we're talking about a blue whale, whether we're talking about humans. We all use DNA and RNA. Pretty interesting. In fact, even viruses, which we can debate whether they're alive or not, retroviruses are relying on RNA in order to mooch off of our other proteins and carbohydrates and living macromolecules. Okay, so let's talk about DNA and RNA. I'm going to try to keep this under 10 minutes, though there's a lot to talk about. So let's just do a quick introduction. So DNA, as many of you know, let's see, we're going to draw. DNA is a double-stranded molecule. And what we have is a backbone, as you saw in that nice cartoon. The backbone is made of a strand like this. Oh no. Okay, still getting used to my new tablet. Sorry about that. It's okay. Okay, let's try again. Okay, so we have a backbone. Okay, I don't know. My tablet is not working very well right now. What a mess. Okay, I'm gonna actually not use my tablet. That's sad. Okay, so DNA and RNA are both nucleic acids. They're both made out of the same monomers, but DNA, unlike RNA, is made up of 2 anti-parallel complementary strands. This is a nice review of introductory biology. So hopefully you all remember this, but if not, let's talk about it. So DNA will have 2 anti-parallel strands. That means that here we'll have guanine, and that will always bind to a cytosine. So G's goes with C's, and then A's go with T's. And this also has to do with how they're made. So enzymes will actually break apart these strands. That's the only time we see single-stranded DNA, when they're being broken apart for synthesis. And every time it sees an A in one strand, it will actually take a T, and the enzyme RNA polymerase will bind that T to the A. And only T's can bind to A's, and only C's can bind to G's in a chemically stable way. And that's because one of the pairs have double bonds and one of the pairs have triple bonds. Now these bonding pairs that make up these double helix, so let's just fill it out some more. Let's say A, T, just give it a random code, A. So just imagine we have this big double helix, all winding, and we have all the base pairs on one side, and the enzymes have done their job, and we have complementary base pairs on the other side. What is the difference between this and RNA? Well, as many of you already know, RNA is only a single strand. That's important. And so we have G, T, C. But one important difference is we have a U. And this is actually how your body can tell whether it's dealing with DNA or RNA. If there's little bits of DNA floating around your body, then your body will actually destroy it because your DNA is inside of the nucleus. DNA is kind of like the master copy of your genetic information. RNA, DNA is used by proteins to make RNA. That's what's actually being sent around your body, telling your body what proteins to make. So we see that RNA has uracil, or DNA does not. So that's how your body tells if it's RNA or DNA. DNA comes in a double helix, as we said. And the last thing we need to know is that DNA has a 5' end and a 3' end, and the double structure is actually anti-parallel. Let me show you a picture of what that looks like, because unlike proteins, amino acids, these other ones, you actually do need to know the details of the chemistry of nucleic acids. So if we look at the molecular structure of DNA, oh, that's a video. I don't want the video, I want the article. Sorry about that. So if you look at nucleic acids, there's a very nice picture somewhere here. Sorry for not having this pulled up. Okay, so here's what you need to know about the DNA double helix. So it's made out of these backbones right here that have a phosphorus, a phosphate group. So that's phosphorus bonded to 4 oxygens, and then it's bonded to a carbon chain. And then we always have this this 5-carbon group, and then it repeats. So this is the backbone of DNA. It always has PO4 bonded to a carbon chain, and then this is just a repeating structure. So if you look at this part right here, this repeats here and just keeps repeating all the way down. And these are all covalently bonded to our A's, T's, G's, and C's. And that's these chemicals here. They're not actual letters. Of course, they represent chemicals. A, G, C, and T are all of these. So this is called the backbone, the phosphate sugar backbone. So this is the phosphate part. That's why phosphorus is essential for DNA. Also, you'll notice that nitrogens are essential, oxygens are essential, carbons are essential. So we're using almost all of those essential elements that we talked about back in video 1. Now, the only difference between the backbone of DNA and RNA, and this one you do need to know, is the sugar chain, saccharide chain That is, in DNA is a deoxyribose, meaning this is a H. And in RNA, the structure is ribose, meaning that's an OH. Everything else is the same. It's going to just look like a big strand of a single-stranded. So if we have a single strand of DNA, single strand of RNA, the only way you're going to tell the difference is instead of having an H here, you're going to have an OH. One of them has an extra oxygen. One of them has been deoxygenated. They both have phosphate, these phosphate group. They both have the chains. They both have the bases. Let's see if there's a nice picture of RNA before we go. Yeah, I don't see it because this is about DNA. But yeah, that's all you need to know. So hopefully that's a helpful introduction. Sorry I didn't have everything perfectly pulled up. But what are the important things you're supposed to take away? DNA is double-stranded. It's anti-parallel. It goes from 5' to 3'. So on one side, we have the phosphorus sticking out. On the other side, we have the carbon chain at the edge. And in DNA, we have the base pairs adenine, thymine, guanine, and cytosine, whereas in RNA, we have uracil. Okay, that's it for today. Have a great day. Thanks for following along in this unit. Okay, and that's it for the video. For the whole unit. Let's go back and make sure we talked about everything. If not, maybe I'll have to redo it. So I hope not. Okay, let's see if there's anything I missed. Okay, so we were supposed to talk about both DNA and RNA have 3 components: a sugar, a phosphate group, and a nitrogenous base. I showed you the picture of that. They go from 5' to 3' with the bases perpendicular to the backbone. Did not use the word perpendicular, so I have to emphasize that in class. I think you could see from the picture though. And then we talked about how DNA is deoxyribose and RNA has ribose. Unfortunately, I didn't have the picture side by side, so I would make sure I do that in class. But the difference is, I pointed out, one of them will have an H group, one of them will have an OH group, because DNA is deoxygenated in the ribose sugar. It's lacking the oxygen. RNA has uracil. DNA has thymine. So the uracil replaces the thymine. That's an important thing we need to talk about. RNA is usually a single strand, as we mentioned. Yeah, so I think we covered everything. But make sure you're always following along in the unit guide. And just like Guy did, this is what you need to know for your test. This is what you need to know ultimately for the AP test. So keep me accountable. Okay, that's behind the scenes and how I make these videos. I hope you enjoyed it. Have a wonderful day. Thanks for following along to the end. One last tidbit, because you're stuck with me to the end. If anybody's still here, I'm going to tell you something about your first test. I'm going to tell you the last question on your test. The last question on your test is going to be this. Very simple. Just going to say, why do you... So it's going to say something like AP Biology, very time and energy demanding class. Why do you want to take it? And of course, I'm not going to mark anyone down for their answers, but I will say this. On the summer homework test, take this question seriously, because I will read it. I will consider why students want to take it. Everyone's going to get a point for answering it, for giving an answer, whether it's short or long. But if you're at the cusp, and what I'm looking for in this question is just this. Why do you want to take this class? Are you someone who is just doing it to help your GPA? This is not the class for you. I had lots of students who are very good students last year get B's in AP, in my AP science classes. AP Science is not a class to take just to bolster your grade. It's not so easy. However, if you're actually interested in biology, you want to go into medicine, you think you might want to use biology or study it in college, and you're willing to put in the work, you're going to make this class your priority, I want you to talk about that. I'm going to take that into consideration when we decide who will be in the class. So make sure you take this question seriously. Obviously, if you do well on the summer homework test, then you don't have to worry about this. You'll be in the class because, however, if any students are, maybe they just are not the best test takers, but they really want to take the class or willing to put in the work, then they belong. So keep that in mind when you see that question on the test. Okay, now that's it. I'm going to run. Have a great day, everyone. See you in a couple weeks.